I think every time before I've been here, it's been in the winter. So I was teasing Neil because he's a snowbird and he flies south. And I was teasing him. We met up in Arizona, and I said, you know, maybe you guys could actually have me in the warm weather sometime. So that's why I'm here now instead of the winter. But now it rains. And it's like, okay, you know, I'm from Arizona. So you've heard about the poor Phoenix resident that was walking down the street one time and a big raindrop hit him right in the middle of the head, and it startled him so much he passed out on the ground, and they threw a bucket of sand on him to wake him up. So, all right, and we'll let you answer that. Which does serve as a reminder. When I was a college professor, I would collect phones. I was one of those, yes. So, the question we're going to ask this evening is, are dinosaur proteins immortal? And then tomorrow night we're going to talk about genetics. And in some ways these dovetail because they're really on opposite ends of the spectrum, and yet we'll see if you come to both, you'll kind of see how they begin to match up, because it covers, it kind of shows the whole spectrum of the controversy of origins and the controversy of just what is it you're being taught in the public system, in the popular views, versus what we as biblical creationists are arguing. Okay, so are dinosaur proteins immortal? Is going to talk about just that, dinosaur proteins, which is an odd topic because 20 years ago they would have said, what? You know, dinosaur proteins, what's that mean? All right, just as a little background, and this is going to start out slightly elementary, but I want to just kind of make sure everybody's up to speed with this. In the 1930s, a man named Charles Lyell proposed that all geologic changes are very slow. And he published his work called Principles of Geology, which Darwin himself admitted was very influential in his thinking as far as the time scale, as far as the changes that occur, all changes are slow. What Lyell did, and others of his peers began then to do also, was they made the assumption that sedimentation, fossilization, canyon formation, river channeling, all of this took millions upon millions of years. Now, they didn't have any analytical methods of measuring this per se. They just simply looked at a formation like this and said, wow, that must have taken a long time. So obviously it took hundreds of thousands, if not millions of years to form. And that then is what he essentially presented in his arguments, was it just takes a long time, therefore everything has to be very old. We now know, of course, that canyons can form very rapidly. We know that sedimentation can occur very rapidly. We know that river channeling can occur very rapidly, so we know the opposite. We know that actually all this doesn't take very long at all. So we see that Lyell built his arguments on a false premise, and yet they became the popular arguments throughout the rest of the 19th, well into the 20th, and even into the 21st century. Often when you talk to someone about the age of the Earth, the age of a geologic formation, etc., you're going to be referred to radiometric dating. Oh, we know because of radiometric dating. We know this is old because of radiometric dating. Well, first you have to recognize that the relative geologic dates of various geologic formations were assigned well before there's any such thing as radiometric dating. So they already had their target dates before there was any kind of radiometric measurements available to use to try to ascertain the dates. We also have to recognize that it really wasn't until the 1950s that actual technology caught up with and enabled measurements of radioactivity such as you could really have radiometric dating processes. Now the question I challenge is, would these methods have been accepted had they not given the anticipated millions of years ages. If they used potassium argon on a particular lava flow that they were already saying must be 20 million years old, and it did not give an age of 20 million, but maybe gave an age of 200,000 or 20,000 or 2,000, would they have said, oh, I guess we misdated? No, they would have said, well, obviously that method doesn't work. See, so you have to understand radiometric dating is not an external verification of the assumed geologic ages that have already been assigned in the 19th century. It is a product of those ages. And if radiometric dating gave older ages, which it often did, then all the more better. We'll take more time, but they weren't going to take less time. So I challenge that when they try to trump this dinosaur information I'm going to give you with radiometric dating, you have to recognize the house of cards radiometric dating actually is. And so I come back and say, let's look at the interpretations that are being applied. All right, the Darwinian interpretation of the geologic record would be that each sediment was laid down much before the next sediment, and these represent vast periods of time from sediment to sediment, and so the changes of the fossils would represent evolutionary change. And so what you would see then, and this would be the standard way to be interpreted, was that this creature then evolved to this, which evolved to this, which evolved to this, and you have the time frame in order for that to have occurred. Now, the flood geology interpretation would be that the flood laid down most of the geologic record, and so for the most part these fossils represent animals that would have been concurrent with each other. So this does not represent vast periods of time, and it does not represent animals that lived millions of years before other animals. It represents animals that would have been simultaneous or would have been living all at the same time. So what we see is two totally different interpretations of the same data, and that's what we commonly get in the arguments of evolutionists versus creationists. Is they use the same data, they give you totally different interpretations. So what I'm going to do is say, all right, let's approach this such that we have creation timeline versus evolution timeline. I do not accept radiometric dating as an external verification of the evolution timeline because it's a product of the timeline. But I will submit that instead, dinosaur proteins are an external verification of the timeline. They came out of thin air, out of nowhere, from a complete side angle that no one anticipated. They weren't a product of anything as far as assumptions or presumptions go. All right, to understand where we're going with this, we first want to step back a little bit and review your anatomy. I'm sure you all had lots of anatomy classes in college and high school and such, but we just need to review it for a moment or two. In bone formation, the first event is that the body lays down a matrix of protein called collagen. Then it lays down on top of that collagen a matrix of calcium and phosphorus, and that gives you the mineralized component of bone. We sometimes tend to think of bone as like a rock, but it's not. Bone may be mineralized, but it's still tissue. In fact, when you're young, your body goes through a process called remodulation, where you actually exchange all of the protein and calcium phosphorus matrix out in all of your bones about every 10 years. As you get older, not so often, and then that's where you start getting older and crankier and just, you know, we all knew we'd age, but we thought it'd take longer, right? Anyway, okay. Understanding then that bone is actually tissue and not just, it's a mineralized tissue, not just a rock, then this is where we're going to go with this, and recognizing then what is the process of fossilization. Okay, an animal dies, bone gets buried, and of course the first thing you want for fossilization is you want burial. The second thing you want is you want burial with a lot of water. Let me think, if I can think of something biblically that might have buried animals with a lot of water. Gee, okay, all right. What happens is the groundwater will bring silica and other geologic materials such as sulfur and iron and carbonate into the bone, and it'll wash out that calcium phosphorus matrix, and it will accelerate degradation and deterioration of the biological material as well. So over time you have then called full replacement of all the biological material, all the biological mineral matrix with the inorganic matrix, the inorganic material from this geologic surroundings. So fossils generally loaded with silica. They also have carbonate, iron, sulfur, and some other inorganics depending on the surrounding environment. Now, my question is just how long does this take? Because what we're going to see apparently is it takes at least 100 million years. Wow, I've seen aluminum foil crystallize in 3 years. I've seen cardboard fossilize in 3 years. So I'm a little surprised that bones take 100 million years. But anyway, all right. In 2005, a scientist named Mary Schweitzer published a paper in the journal Science. Now, let me caveat this for a second. This is called the seminal paper on this topic, but it's actually not the first paper. There are papers scattered throughout the scientific literature dated all the way back to 1923, where scientists were reporting the possibility that it looked like maybe there was some red blood cells, maybe there was some blood vesseling, maybe there was something still biological in the dinosaur fossils. But for the most part, those usually were disregarded because it's very easy to just say, well, it was contamination. It was often published in a kind of an obscure journal or maybe a very specifically disciplined journal that other people weren't really reading and catching their attention. And so what we see is the 2005 paper that Schweitzer published is the one that really caught people's attention. Now, it was published in the journal Science, which is the second most read scientific journal in the world, and that might have something to do with it because now it caught the attention of a lot of people besides spatialists in geology and paleontology. But also, this can't discount the fact that it published color pictures. Never underestimate the power of color. Up until this time, most journals published black and white simply because of the expense. But by the early part of the 21st century, the expenses were starting to come into coordination enough, shall we say, that began to publish more color. And they were also starting to publish material online, which would then, of course, contain the color as well. So there's any number of reasons why this 2005 paper may have caught the attention that it did, but it definitely caught people's attention. She also quickly followed up with 2 papers in 2007. And in all these papers, the argument was that she had a femur from a T-Rex that she was finding soft, pliable, stretchable material from, and also finding fragments of what they were identifying as sequences from the protein collagen. So in other words, there's still biologic material in this T-Rex femur, even though they were saying that it's got to be 68 plus or minus 68 to 70 million years of age. Of course, the question was, how in the world can you have a fossil that old still contain biological material? And that's still the question even today. So my challenge is the presence of this material is a direct biochemical challenge to the assigned dates of the fossils. Now, here's the dilemma for the evolutionist community. If dinosaur fossils are dated wrong, that means all the fossils are almost certainly dated wrong. If all the fossils are almost certainly dated wrong, that means all the rocks are dated wrong as well. And the whole dating scheme comes crashing down. That's why it's very important for them to prop this dating scheme up, including then they've got to address the presence of this tissue. All right, so this then introduces where the Creation Research Society comes in. By the way, if you're not familiar with the Creation Research Society, it is the creation society for young earth biblical creationists. It's an international society. As a society, then anybody that subscribes to our position statement can become a member. Okay? Our primary focus is like with any scientific society, it's research, publication, and conferences. By the way, Walter, you got to come to some of our conferences sometime. All right. Anyway, that's our website, creationresearch.org. we decided we wanted to also start participating in the soft tissue research, because not that we were saying that it was being done wrong. In fact, we say that Mary Schweitzer's done some very good work. But we just didn't like the idea that the evolution community would set the tone and the direction of all the research. We wanted to also be participating in the research and be able to do some of what we felt they might not be willing to do. All right, in 2012, the Creation Research Society sent a team to eastern Montana in what's known as the Hell Creek Formation. The Hell Creek Formation in eastern Montana is a treasure trove of dinosaur fossils. In fact, it's where Mary Schweitzer and Jack Horner found that T-Rex femur. It's where a lot of other soft tissue fossils have been found. So we went to that region to find some fossils that we could work on as well. Okay, this was the first one we found. This is the brow horn of a Triceratops. In ground, it measured 46.5 inches in length. As you can see, though, the end of it is extending out the surface and is obviously heavily weathered. So the overall original length of this horn probably was 50 inches or more. You think of that. Can you imagine the size of the head that that horn was on? This was a great-great-great-grandpa Triceratops. This is a beast. The museum people that were serving as our guides were just beside themselves, because they had, as far as they knew, this was the single largest Triceratops horn found in eastern Montana, if not all of Montana. And of course, to them, this was going to make a beautiful museum display, but that's not what we were there for, because science has its prices. And we were going to take it, and the first thing we did was break it up. And that's, in fact, its current situation. This is broken up in little pieces. Now, some things to notice about this. First, it's about a foot down from the surface, but it's encased in sandstone. And that still took 3.5 hours to get to that, but it's only about a foot down. That means there's really no thermal insulation. So whatever the temperature was outside is what the horn experienced. And any competent biochemist can tell you one of the fastest ways to deteriorate biological material is to heat it up and cool it down, and heat it up and cool it down. And eastern Montana, I'm not sure quite how hot it gets here. Okay, I assume you hit 90s from time to time. All right, and then eastern Montana, I mean, I think minus 300 probably sometimes, I don't know, but anyway, they get really cold sometimes. So what you have is this humongous shift in temperature, which is one of the worst ways of trying to preserve tissue. Also, it's close enough to the surface that it had infiltrating ground rainwater. And in fact, when we pull this up, it had water puddled underneath it. So it's damp. And it was muddy. And I was sitting in the truck as we were driving out of the field, and I turned to my colleague and I said, there's no way we're going to find anything in this thing. It's just, the state of it is just so horrendous. I got looking a couple months ago and realized that that picture is all over the internet. Now, most of the internet sites that have that picture don't know what they're talking about. They're talking about all kinds of aliens and everything else, but that picture is all over. So my arm is all over the internet holding the tape measure. When examination of the horn began, this is what was found. And Mark Armitage, who was doing our microscopy, started peeling off layers of tissue. If you'll notice, now this is a static picture obviously, but that's a piece of tissue. It's stretchable, it's pliable, it's squeezable. The little dark spots in there, those are bone cells. Those are dinosaur cells. If we look at it closer under electron microscopy, we see that specifically these are osteocytes. Osteocytes are unique shaped bone cell. That's how we know they're osteocytes. It's not contamination because skin cells and other kinds of cells look totally different. Also, see where the arrow was pointing, that's pointing to a structure on the osteocyte called a filopodia. Filopodia are very, very thin protein extensions with a membrane covering, extremely fragile. And yet there they are, still well preserved. So you're saying that this horn sat in the ground for these tens of millions of years And these extremely fragile Philopodia just continued to pluck away. See, that already biologically is inconsistent with the assigned age of the horn. Now, a few years later, an intern in a museum wanted to learn about soft tissue, so she came down to my lab. She brought with her the end piece of this Thesaurus vertebra. The story behind that is that the museum dug up this vertebra column, but the end piece there at the bottom left was so damaged that they didn't have any use for it. They couldn't reconstruct it. They couldn't salvage it. So they just simply put it in plaster, wrapped it in aluminum foil, and set it on the shelf. So again, no spatial preservation. She brought it with her in part just as a fossil to examine. I had some fossils too, but she brought this with her just say, oh, I just want to look at it too. So she was one day just scraping away on it under the microscope, and I told her to stop. I turned the camera on. And then we washed it off some, and she did this. So you could almost just throw the thing on the grill. Real dino burger. Now, clearly, what you see there is, first off, not a figment of your imagination, okay? And it's not some kind of contaminating. A bird did not, drop part of its muscling or something down into that fossil. This is straight from the fossil. Other studies, for example, a study in 2015 in the journal Proteome Research isolated blood vessels, and in the blood vessels they did a protein analysis, and they found not only collagen again, but they also found tropomyosin, actin, histones, tubulin, and myosin from the femur of a duck-billed dinosaur, so a different dinosaur. So already we've now got 4 dinosaurs, just what I've talked about, that we've found that have had tissue found in them. 2 in my lab, 2 in Mary Schweitzer's lab, and that's just the tip of the iceberg. Now here becomes the problem for the evolutionist community. Studies of collagen degradation say that the optimum condition, collagen would last no more than about 2 million years, probably less. If you're finding fragments of collagen still in dinosaur fossils that are supposed to be 70, or as with the duckbill, 85 million years old, and of course now they have found fragments of collagen in a dinosaur fossil dated at 195 million years old, and they have some biologic material they removed from a beardworm dated at 550 million years, you begin to see there's a problem here. There's a major problem here. And biologists either ignore this or they say, okay, I can't believe it. Because any competent biologist recognizes the two don't go together. You don't have 195 million year old biological material. Interesting, as a criticism of Mary Schweitzer's 2005 and 2007 papers, some biochemists wrote a response and published it in the Science Journal in 2008. And in that they said, okay, we calculate that at 7 degrees centigrade, collagen has a half-life of 130,000 years. And you can take that half-life and you don't go very far before you recognize you're not getting to 65 million years. But they said here is another problem, and this is part of the conundrum the evolutionist community faces with their time scale. By their own time scale, at the time the Hell Creek Formation would have been forming, It was what would be considered a megathermal environment. In other words, greater than 20 degrees centigrade, which accelerates degradation. And they pointed out the half-life would have only been 2,000 years. So they are saying, and to this day, the authors of this criticism are still maintaining their position. There is no way there is biological material in these fossils that is original. Now, they're in a situation, they either have to reject the date or they got to reject the biological material. Well, they reject the biological material, they're not going to reject the date. In 2017, the same group of biochemists published a paper where they said, you know, here's the problem we think is going on. Most of the collagen being reported to be detected in the dinosaur fossils is really just one sequence, which is actually an ostrich collagen sequence. So we think everything's been contaminated with ostrich collagen. Apparently, and again, I'll give Schweitzer credit for this, apparently they had already anticipated this criticism because within 3 months, they published again in Journal Proteome Research where they detected 6 additional collagen sequences in the femur of the duckbill, and these sequences actually correlate closer to crocodile collagen. So I guess the dinosaur fossils being contaminated with both crocodile and ostrich. I guess I don't know if the crocodile was chasing the ostrich, I'm not sure, but anyway. So that kind of undercuts their argument, and these group of biochemists haven't responded yet to this. Also in 2011, in a PLS1 article, the researchers are noting that they were finding what they identified as native collagen fibril structure that had the typical perpendicular alignment that you're going to find if it's native collagen. Okay, they said contamination would likely give you a random alignment, not that specific perpendicular alignment. They then went on in the publication to say, quote, contamination would not be expected to be overrepresented in regions that so well reflect collagen fibril structure function relationships in native vertebrate tissue. In other words, if it's contamination, it certainly is not behaving like contamination. It's behaving like you would expect native collagen fibril structures to behave. Okay. Another ding at contamination comes with, as technology continues to advance, we are now analyzing dinosaur fossils with a system called Fourier transform infrared spectroscopy. Okay. Typically just abbreviated FTIR. And it will provide an infrared spectrum of the bone. All right. What it will do is it will allow you to identify the chemical characteristic of amino groups that are going to be unique to proteins. And since type 1 collagen is virtually all the protein you're going to find in these specific segments of the bone, then for the most part we assume if it's a protein, it's type 1 collagen. And so of a 195-million-year-old sauropod rib, FTIR analysis found that in fact there was still protein. Now, you can't say this is contamination because this was without any kind of extraction, any kind of handling of the fossil whatsoever other than the careful handling of it to put it onto the machine. So it wasn't like some kind of contamination was introduced into the fossil or introduced into the analytical machine. Okay, this was also done with a crustaceous bird where they again found intact fragments of collagen protein without any kind of handling or extraction processes. So in other words, this is still it in its native state. Okay, there's also what's called second harmonic generation imaging. See, as technology advances, see, people accuse creationists of not wanting science, which is the opposite. We love science because one thing science does is it keeps vindicating us. It keeps vindicating us. And Walter back there can say the same thing. As they've dug into the human genome, it has so vindicated and validated everything creationists have been saying. So we love science. Keep digging, keep digging. All right. This picture was given to me courtesy of my colleague Brian Thomas, who's been working with me on some of this stuff, which, by the way, as we speak, Brian is at an ancient protein conference in Copenhagen. So I'm in Minneapolis, he's in Copenhagen. I'm not quite sure how that worked, but anyway, so with second harmonic generation, what you're able to do is the red specks of this analysis of a fossil. In fact, it's a Triceratops femur. The red specks are collagen. And when you then overlay it on top of the fossil, this shows you the pockets of collagen still in that fossil. So this is not contamination. This wasn't introduced as part of an extraction process. This wasn't some contaminating material still lingering in the mass spec or in the GC or anything like that. See, so the argument that what's being detected as contamination is steadily, dramatically being eliminated from the scene.
Definition on the half-life that you talked about, 2,000 years. What does that mean, half-life? That it degenerates or disappears? Or?
It disappears, right. In other words, 2,000 years, under the condition they were talking about, in 2,000 years you'd have half of what you had originally. And then 2,000 more years half. So half-life would be the same as you'd give definition for a radiometric.
And then it's replaced with material?
It's going to be replaced with the exterior inorganic minerals, the silica, the carbonate, the sulfur, the iron, depending on what's in the surrounding environment. Okay. Other studies done by other labs, and you have to recognize as well, it's not just us and it's not just Mary Schweitzer. There's a lab in Poland, there's a lab in UK, there are labs across the world working with fossils across the world. I still get responses, people trying to tell me, well, it's only been Mary Schweitzer that's found it on one fossil and she's recanted. Okay, what planet are you on? Because that's not Earth. Okay, it's multiple laboratories working with multiple fossils found in multiple continents. Pliable tissues found in a variety of dinosaur fossils. I think it's up to, and I don't, there's other creationists that keep a list. I don't keep an accurate list myself. But as far as dinosaur fossils, I think the list is almost up to 40 different fossils now. Okay, morphologically distinct cells are present in the tissue. Collagen fragments are frequently detected, but also other proteins, actin, myosin, tubulin, are frequently found as well. Now, speaking of which, evolutionists love to try to say, well, maybe under a certain circumstance, collagen, which is a relatively structurally stable protein, that's why it's used as a matrix for your bones, okay? Collagen maybe somehow could have survived for all these tens of millions of years. They almost never, even though I continue to challenge them, they almost never want to talk about actin, myosin, tubulin, etc., because they know they can't. All right? Actin and myosin, for example, undergo very rapid postmortem degradation. If they didn't, you would not eat steak. It'd be too tough. Right? Osteocalcin, which has not yet been found in dinosaur fossils, but represents another bone protein, we see how little would be left after 65 million years at 10 degrees centigrade. In other words, it'd be beyond our detection levels. Okay? Albumin, which has also not yet been found, but is more stable than many of the proteins that have been found, 100,000 years is considered the extended life of it. The reason I don't give tubulin and tropomyosin and such, because nobody's determined the half-lives of those, because nobody's worried about it, because it's so short. So the fact that those are being found and the fact that the evolution community doesn't want to talk about it is a very telling story. Okay, so in an interview a few years ago, Mary Schweitzer gave a good summation. I think this is a very good summation. So that leaves us with two alternatives for interpretation. Either the dinosaurs aren't as old as we think they are, or maybe we don't know exactly how these things get preserved. Fair enough. Fair enough. Here's the problem. See, that's not even an open topic. You can't challenge the age of the dinosaurs because they're so solidly dated by all the radiometric dating. That's the response I get all the time. It's irrelevant what kind of tissue you find. It's irrelevant what kind of material you find. They're so already, we know how old they are because of radiometric dating. I say, but I don't buy the assumptions you're putting in radiometric dating. If you know anything about radiometric dating, you understand when I say parentless argon. The moment I found out and studied about parentless argon, I said, well, that blows the whole thing right there. Because if you admit there's parentless argon, you admit that your assumptions are wrong. Because if your assumptions are right, there should be no such thing as parentless argon. Here's the deal in radiometric dating. If rocks really are 100 million years old, 200 million years old, 300 million years old, a little bit of initial material would be irrelevant. That's why they think they can dismiss parentless argon. But if they're what we're claiming, 4,000, 5,000, 6,000 years old, a little bit of daughter compound to begin with blows the date way out of the water. All of a sudden you take a 6,000-year-old rock and you make it 60 million years old. All right, so how does biological material survive in a 65 million-year-old fossil? Of course, I say it doesn't. But clearly the evolutionist community has their answers. Okay? They're not just, rolling over, obviously. All right, the iron model is by far the most popular. Now, they're not saying this is the only preservation process going on. In fact, see, the more they can appeal to, then the more they just, they can snow you under this and this and this and this. Okay, it doesn't make any difference. If any of them work, they can just sound good by throwing out all these possibilities. You got the iron model, you got aluminum, you got pH 5, you got all these that throw out. None of them have been tested, but we speculate they could work. All right, the iron model is by far the most popular. In fact, in the new Jurassic World movies, the iron model is what's being referred to. See, used to be they got the DNA from where? Remember? Yeah, from amber entombed mosquitoes, and they would pull the supposed dinosaur blood out of the mosquito gut and extract DNA from that. Of course, found out that doesn't really work. Okay, but of course it was science fiction anyway, although when I was teaching, I was a university professor at the time the first movie came out, and unfortunately when it comes to university students, science fiction and nonfiction seem to kind of merge, and they struggle to separate the two. So anyway, but they found that amber entombed mosquito doesn't really preserve very well. So now in the new Jurassic World movies, they're getting the DNA from where? Iron preserved dinosaur tissue. See, so that's how it's getting into the popular psyche through these types of avenues. All right, the argument is that iron stabilizes tissue. Blood contains iron in the hemoglobin. So the blood then is going to be, and I put a question mark because they're going to argue there's a high concentration of blood in bone, and that's questionable how much blood you're really going to have in a given bone. Okay? And so the blood in the bone will preserve the biomaterial because of the iron it contains. Okay? That in a nutshell is the basic assumption of the iron model. Now the iron model works using a process called Fitton chemistry. Okay, Fitton chemistry says we're going to take the iron from the hemoglobin, maybe some contribution from myoglobin, which is going to be a similar protein in muscle cell. That iron will catalyze a series of reactions. See, iron loves to catalyze reactions. If you took a piece of iron and you put some water on it, left it for a couple of weeks, what would happen? Yeah, you get rust because the iron accelerated oxidation. The iron accelerated some reactions. All right, so the idea is iron will accelerate and catalyze reactions that will result in the production of ions and what are called free radicals. Now, you recognize an ion will have either a positive or a negative charge, and it'll seek its opposite charge. So a positive charged sodium will seek a negative charged chloride, and you put the two together, you get sodium chloride. But a free radical doesn't care about charge. We designate free radicals with the dot. So you got the HO dot, that's the hydroxyl free radical, highly reactive molecule. It doesn't care about charge. It just reacts with everything. If this is loose in a living system, it can become potentially very dangerous. That's why your body has a lot of enzymes that work to clear these things out. Of course, in a dead bone, obviously you're not going to kill it, it's already dead. So the idea is that they'll say it can now be free to engage in reactions, and these reactions are potentially going to help preserve the proteins. One of the reactions that the free radicals engage in is going to be cross-linking of the proteins. Okay, here's what will happen. The circles are amino acids, and this is supposed to represent then a peptide strand of a protein. Okay, the free radicals react with the two of the amino acids. The reaction will drive them then to form a cross-link with each other, and it essentially will cause the protein to start knotting itself up. If you think of the protein as a string, the more you knot it up, the tighter it gets, then the more resistant it becomes to enzymatic and microbial degradation. And that, in essence, very simply, is the iron model. Let's wad these protein strings up real tight by cross-linking so they become less available for enzymatic and microbial degradation. Everybody follow that so far? That's the iron model in a nutshell. All right, now what they don't typically tell you much about is there's a lot of other reactions being catalyzed as well that are destroying the protein at the same time. So it becomes a race. Do we form the cross-links to preserve the protein before we destroy the protein from the very same reactions? Okay, so there was one and only one experiment that's been done so far to try to verify the ability of iron to serve as a preservative. This was done by Mary Schweitzer's group in 2014, where she took ostrich blood vessels and soaked them in hemoglobin or in water, and then visually analyzed the results. How quickly do they degrade? Her reported results were that the water-soaked blood vessels showed significant signs of decomposition within 3 days, whereas after 2 years, the hemoglobin-soaked blood vessels still were intact. So she did the math and she said, we have a 240-fold increase in stability. All right, so then if you look at it and say, okay, if we get under ideal conditions 2 million years worth of preservation of the collagen proteins and multiply that by 240, we get plenty of preservation, so we can go on, we can get way on past the Precambrian even. So the idea is that this answers it, we're done, case closed, move on, creationist 0, evolutionist 1. That's what they want you to think. All right, here is some pictures of what they published as their results. Now these are in black and white, be a little more revealing if they were in color. These are in black and white. The hemoglobin-soaked blood vessels versus the water-soaked. Just looking at that, we notice clearly that the hemoglobin-soaked are much darker. If this is in color, they'd be a bright red, because hemoglobin, when it's in the presence of oxygen, has a bright red color. If you go to the grocery store, do you buy the brown meat or the red meat? See, the grocery store knows that. They're packaged in plastic material that is permeable to oxygen, so you get plenty of oxygen into the meat, so it's nice, bright red. So when you're doing a visual observation, see, we got a nice bright red, that looks like that's well preserved. If you put a piece of meat in water for a couple days, what's going to happen? It's going to bleach, it's going to pull the color out, and it's going to look like that piece of meat you're not going to buy. And so we see with these blood vessels, well, they're pale, they're anemic, if you will. They just don't look as lush. So visually, subjectively, we have a difference just simply based on that. Has nothing to do with deterioration, it's just based on visual appeal. Bright red versus bleached pale. Okay, but there's other challenges I would pose as well. Now, not to trivialize a 2-year study, because in science, a 2-year study, that's a third of your tenure track. I mean, that's not insignificant. But we have to recognize a 2-year study at constant temperature is not very readily extractable to 65 million years at fluctuating temperature. There's only so much you can say about a 2-year study. Now, the last I heard, she's still saying that the hemoglobin-soaked blood vessels, after I think 6 years or so, are still looking fine, which they might be. I have buffer-soaked blood vessels after 3 years still looking fine. No hemoglobin in them at all. Okay, the study admitted that they couldn't get the red blood cells to sufficiently lyse on their own. So they had to mechanically disrupt them to get them to release the hemoglobin. Because if the red blood cells don't release the hemoglobin, the iron won't be able to get in contact and react with the blood vessels. So by their own admission, they could not naturally duplicate, what they're claiming is happening. So already we have a problem as far as the study really offering validation of the iron model. Also, water is a very harsh biological material, very, very harsh biological control. Water is considered a biological solvent. That's why you see if you take that piece of meat and put it in water in a few days, it's nice and bleached and starting to come apart because water dissolves biological material. Proteins and carbohydrates will dissolve a lot in water. Lipid doesn't so much, but the lipid's not what's holding the biological material together. It's the proteins and the carbohydrates. So by using water only, she was creating a dramatic difference. I'm not saying water isn't appropriate as a control, but it's not appropriate as your sole control. She needed to use a series of biological buffers as well. Like I say, I have blood vessels soaking in biological buffers that are doing just fine after 3 years. Why didn't she do that? Because when you put it in water, you're maximizing the likeliness you're going to get rapid degradation, because that's what water does. Your body is loaded with water, but it's not really in a water state, it's in a biological buffer state. Because if it was in a water state, you'd be dissolved on the floor. Okay? The results that were reported were strictly visual, and I think this is very telling. They were strictly visual. They threw the vessels under a microscope and looked at them. The problem with visual is you tend to find what you're looking for. So if I'm looking for degradation, I'm going to find it. If I'm not looking for degradation, I'm not going to find it. Okay? They claimed that, for example, the water-soaked vessels had a much more rapid rate of vessel wall degradation, but they offered no measurement system or protocol of measuring rate of the degradation. It's strictly subjective. Why was there more analytical processes performed? Why was there not more analytical processes performed? And the answer is, I don't know. I can be cynical. I'm not trying to be cynical because Dr. Schweitzer has done some very good work, but I could be cynical and say maybe it's because they didn't want too analytical of result. So we're going to do the analytical. It's part of what Brian and I are working on. Okay, most dinosaur remains are found highly scattered, that is disarticulated. In other words, you'll find a single bone here and a single bone here and a single bone over there. That's why finding 75, 85% of a dinosaur skeleton is worth millions of dollars. So if you find one of those in your backyard, you got it made. But if you have single bones scattered, then this is not a bloodbath. These bones are not sitting there soaking in blood. So one of the questions is there really enough iron in the blood? And this was raised in a 2016 paper where they challenged that they don't think there's enough iron in the blood. So we're going to have to at least supplement with external iron, get it from the environment. And yes, we recognize there's iron in the environment, recognize that fossils have iron in them. The question is it going to be able to get in fast enough to give preservation? Okay, well, the problem you face in doing that is geologic iron is very insoluble. In fact, you need low pHs. You need pHs approximately pH 1. Be like sticking your finger in hydrochloric acid. So if you have this very low pH necessary to solubilize and bring this geologic iron in, it's going to enter the bone very slowly, going to precipitate very rapidly, and it's then going to be a contest. Can you get enough in before the tissue is degraded, and can you get enough in to protect it from the pH 1 that's being used to bring the iron in to begin with? See, so we have a major problem. And it's not just me saying this. This is in the literature that is being discussed back and forth by different scientists of we have a problem here with the iron model because we don't know where to get the iron from. It's a problem trying to secure enough iron for the model to work. Okay. This brings up the question then just how reliable are the protein decay studies? Remember we were talking earlier about that studies were showing that collagen will degrade within 2 million years even under ideal conditions, that actin and myosin degrade very rapidly, that osteocalcin will degrade in 100,000 years. How reliable are those studies? The Creation Research Society published the iDino issue, which, where'd it go? Here we go. This was published in 2015, and this is a technical report of some of what I've talked about. Okay, but we've gone past that now, more recent. But this is a technical report. This is a creationist response to the topic up at that time, talking about the tissue, what's in the tissue, talking about the fit and reactions of the iron model and such. Fazell Rana, who worked for Reasons to Believe, which they identify themselves as creationists, but they are old Earth. Okay? And I can't seem to get them to understand that when you are old Earth, you have to recognize that much of the method and the methodology used to date is evolutionary biased. You say you reject evolution, but you accept the tenets of evolution. I find that in conflict. But anyway, he wrote a book called Dinosaur Blood in the Age of the Earth, and he argued that the thermokinetic studies used will alter how collagen decays. Now, I have to speculate that our spatial idino issue stimulated the writing of this book because we're cited in it so many times. And Brian and I were laughing at each other which one of us was referenced more, him or me, because we're the two prominent ones in this book, and then Mark Armitage is a little bit less. Okay, so the three of us, we are featured a lot in his book. But one of his big points is the thermokinetic studies aren't doing it right, and therefore they're creating false data. So the lifespan of collagen is actually much longer than what these kinetic studies are showing. That was his argument. All right, so I wrote an article that was published on the Answers in Genesis website, and I challenged him to provide experimental support for his assertion, because this was just an assertion. Okay? Show me experiments that show this, because thermokinetic studies have been done by biochemists for decades. And you're now questioning the validity of these. Okay, well, give me a study. All right, so I got not only one, but I got two responses, so I felt really privileged here. All right. One of the things I love about his title is, Does Dinosaur Tissue Challenge Evolutionary Timescales? But I thought he was a creationist, so why would he care? So is this maybe kind of a subconscious admission that even old Earth creationists accept the evolutionary timescale based on evolutionary assumptions? I don't know. I was just intrigued by his title. But in his response, he argues that there's a 1972 study that shows that collagen will denature at a temperature that's far below the temperatures of thermokinetic studies use. All right, let me explain to you for a second a thermokinetic study. If I took collagen and put it in a buffer and set it on the shelf, my great-grandchildren would be the ones analyzing the degradation it would take years and years. So what you do is you heat it up, and you remember your chemistry, you use heat as an energy source that drives reactions faster, so that drives degradation faster. Not a frequent temperature, not the only temperature, but a frequent temperature used in thermokinetic studies for protein degradation, especially collagen, is 90 degrees. And that's 90 degrees centigrade, not 90 degrees Fahrenheit. Big difference, as one of my lab technicians found out. Okay, not the same thing. So his argument is that 90 degrees is actually going to denature differently than lower temperature. So what he's saying, so what we need to understand here, the difference between denaturation and degradation, as I'm defining it. Okay? Denaturation is where you break the sulfur and hydrogen bonds, inner bonding between the strands, so that it loses its tertiary and secondary structuring and just simply now becomes its primary structure, which is the sequence of amino acids. Degradation is where you now start breaking the peptide bonds themselves. You just have individual amino acids. See, so there is a difference between the two, but you haven't really degraded the protein until you've broken the peptide bonds. So his argument is that because the denaturation temperature is so much lower than 90 degrees, when you use 90 degrees for your study, it's going to denature very quickly, and therefore it's going to accelerate degradation as well, because now it's going to undergo a degradation rate different than the degradation rate it would undergo at a lower temperature. So his argument is that you no longer now can make the proper mathematical conversion because it's not degrading in the same manner that it would degrade at a lower temperature. In other words, the temperature of the bone in the ground. Okay, that's in essence his argument. All right. We have what I call Rana's mistake. Actually, there's two mistakes. One of them is not so much a mistake as it is an assumption. The truth of the matter is, if we go back to this slide for a second, no one knows this. This has never been verified. Because in all honesty, probably to this point, biochemists didn't care. But no one's really verified if there is this disconnect between the temperature range of the denaturation versus the temperature range of the degradation. But what's actually his mistake is the 1972 study that he cites as his experimental basis used muscle collagen. But the studies that I am citing use bone collagen, which is mineralized, and bone collagen has a denaturation temperature close to 90 degrees. See, so all of Rana's assumptions then are for the most part irrelevant because it doesn't degrade the way that he's thinking it degraded because it's bone collagen, not muscle collagen. Okay, Jeffrey Botta, who's an organic geochemist, in 2006, and I've not heard anything from Jeffrey Botta since 2006. Okay, in 2006, in an interview, he said that he cannot imagine soft tissue surviving for millions of years. Has nothing to do with the biochemistry rate of protein degradation. It has to do with radioactivity. He says even if T-Rex had died in a colder, drier climate than Hell Creek, environmental radiation would have degraded its body. Bones absorb uranium and thorium like crazy, and we know that. We can take a dinosaur fossil, put a guard counter over it, and it lights it up. That's very common. Okay? You've got an internal dose that will wipe out biological molecules. So he's saying you cannot have a fossil buried in the ground for 70, 80, 100, 150 million years and have any biological material left because it's going to be so prolongedly exposed to radiation it'll just wipe out. Even if nothing else did, the radiation would. And there's one thing I've noticed. There's no preservation model that addresses this, and there's no evolutionists out there right now that's even talked about this. I've raised it several times, and I get ignored. In Rana's book, he quotes me as talking about this, but then he doesn't offer a solution, which I thought was a little odd. So I would say if all else is still not tickling your gizzard, shall we say, this is a death knell. You cannot preserve from tens of millions of years of exposure to radiation. It just simply doesn't happen. Okay, so the dinosaur tissue proteins is not consistent with the assigned ages of the fossils. Instead, see, it's consistent with the biblical creation model, where the fossils instead will represent ages of some 4, 5, 6,000 years. In other words, ages represented in terms of thousands, not terms of tens of millions. And just some resources. Ross already pointed these out a little bit. The Echoes of Jurassic video, which is our video presenting this material in a very basic form. This is the video you want to show to your friends who have not really heard about this before. Then I have a book, Echoes of Jurassic, which goes into much, much more detail. So if you're ready for a little more meatiness. And then the Creation Research Society Quarterly iDino report, that's much more technical. So you kind of take them in turn. The video is much more popular, the book is more technical, and the quarterly is very technical. So you can wade through all three of them then as you step up in technicality. Okay. Question here.
Yeah, so in your Hell Creek discovery of the Triceratops horn, how long would it take radiation and limestone to degrade the collagen left in that horn specimen?
Did everybody hear the question? No. No? Always a troublemaker. The question, which I really can't answer, is for the Triceratops horn that we found, how long, sitting in that particular environment, how long would the radiation have taken to destroy it? And of course, the reason I can't answer is because you'd have to actually go in and measure the amount of radiation in that specific area, which we didn't do, although it might be interesting to do that sometime. But clearly, what Botta was doing, of course, was generalizing, but he was saying that wherever you are, the radiation would be sufficient that you can't get away from total degradation, you know, in the lifespan of what was supposed to be the fossil's history, you know, 70, 80, 90, 100 million years. So it would be enough that you may argue, well, 10 million years, it might still be 20 million, but his argument is 70, 80, 90 million, it doesn't make any difference. You have enough worldwide radiation that would still take care of it. Yes?
Could you share what happened to Mark Armitage after he published what he found?
Well, to keep it real short, and of course you'd have to ask Mark himself. He'd be the one to tell you the story. But the gist of it was that there became members of the faculty at the university where he was working that just didn't like a creationist working there. And they took grounds to release him. And they dismissed him. And so he filed a lawsuit against them, claiming prejudicial dismissal is what it came down to. And the judge didn't make a final ruling because what happened is the judge did not dismiss the case. And in not dismissing the case, then the university, the state, decided they weren't going to pursue it. So they went ahead and paid, X amount of money to him then. Yeah.
You see that happening around, persecution of Christians?
Oh, persecution's very common. Very common, yes. And unfortunately, it's getting worse. When I have debates with evolutionists, they want to deny it all the time because, they're just not a good scientist. They're just doing their work. Because they know that if they stood up in front of an audience, basically an audience of taxpayers, and said, yeah, we dismiss any creationists even if they're doing good work, they know that would sound pretty tacky. So they want to, well, that doesn't really happen. But of course it happens. It happens all the time. It's been documented several times over.
But the way this method of determining age, by what, how preserve structure, wasn't to use a scientific method to determine the age of the things, for example, dinosaur, chicken.
How they have currently established the age did not use this. Right. You're saying they did not use a objective method to determine the age. Yeah, and we would agree. Yeah, I would agree with that. That it was really originally based on assumptions, and those assumptions then have carried through. So the assumptions then become the filter to determine if another method like potassium argon is working or not. And if the potassium argon doesn't give them the date that they want, then they obviously use a different method. And creationists have written about this a lot. Obviously it gets ignored, of course, but written about this a lot. The Skal 1470, or I'm sorry, what's the Skal, is it 1470? Yeah, all right, for some reason I'll tell you. The Skal 1470 is an excellent example where the lake bed had already been dated. But then when they found this fossil, that gave it too old of an age, so they threw out all the dates. But see, that's supposed to not happen because the lake bed's supposed to have been securely dated. So it proves that the dating is based ultimately on the fossils, not on the radiometric dating.
Okay, I'm going to interject. The question that was asked about is there documentation about persecution of creationists? Yes, there is. I believe his name is Jerry Bergman.
Jerry Bergman has written several books, yes.
He's written several books of which we have, thick, 2.5-inch thick books of documentation of which we have at the CRC Lending Library.
The question is, of the samples, the fossil samples that are taken to research the dinosaur proteins, how many of them turn out having nothing in it versus how many have actual proteins in it? In other words, do you have hundreds of bones that you had to throw out before you found something? Or is this very common and part of the fossil record that dinosaur proteins are pretty much all throughout the fossil record?
I'll give you a two-part answer. First off is I can't tell you the percentage because nobody's keeping track and reporting on the fossils that they don't find tissue in. But a 2015 article in Nature Communication referred to tissue found in dinosaur fossils as common. So it's common enough that finding it is no longer considered a surprise. I forgot. Did everybody hear that? I forgot to turn my mic back on. Did you hear that in the back?
Yep.
Okay. This is.
But do you find similar biological material in bones that are not dinosaurs?
Yes, yes. I tend to focus on dinosaur because that's what gets people's attention. But yes, if you take if you take the list of Cretaceous, Jurassic, Triassic, etc., fossils as a whole and not just dinosaur, the list gets probably, I think, to over 100 different fossils that some kind of biological material has been found in, including Cambrian fossils. See, that's 500 million years old by the popular date.
Do you happen to know how far your Triceratops horn was from that T-Rex Schweitzer find?
About 60 miles. Yes.
Did you say that collagen samples have been found in dinosaur bones all over the world, Poland?
Yes, yes, yes. Several different continents by several different labs, right? Not just Mary Schweitzer, not just one or two fossils, but fossils found in the U.S., fossils found in China, fossils found in Poland.
Are they usually found in just sandstone or any other kind of fossil?
There are some reports of not necessarily dinosaur fossils, but other fossils not found in sandstone that do contain tissue, yes. Yes?
Is it true that most likely big bones would be more likely to contain soft tissue? And if so, maybe they haven't found more because the big bones are the ones they want to preserve because they're worth something like the one you found, you know?
Certainly, you're reluctant to break up a big bone because that is a valuable find. And some paleontologists have criticized Dr. Schweitzer for even, doing it to begin with. Why would you break up? But see, they had to break the bone to begin with in order to lift it out of where they were finding it because it was down in a gully area, down in a ravine. But the irony is that, like that vertebra, the broken vertebra that I showed you the video from, see, the pieces that were brought by that intern, they weren't very big. Now, the bone itself was bigger, but the pieces were fairly small. Yes, we would assume that big bones may have a better chance of preservation simply because they would have more tissue to begin with. But smaller bones have been found to also contain tissue. Yeah?
Is there certain rocks that bones get preserved in better? Like, is that sandstone? Is there other certain parts, things that they're buried in that help preserve?
I'm sure that someone else could give you a better answer than I can because I'm not a geologist. But clearly, sandstone is going to be a good geologic situation for fossilization. But that's now you've gone beyond me because now you're moving into areas that I'm a biologist. I'm not a geologist. Yes?
Could there be lots of fossils that do have soft tissue in, but no one's going to find them because they're not going to look?
Oh, absolutely. And that's clearly been done for years. No one was looking. Why would you look? And the few times, as I said, the few times that people did look and did report, it was for the most part ignored up until 2005. Even Dr. Schweitzer reported has a couple previous reports where they hint at it looks like there's maybe some red blood cells here. It looks like maybe there's something biological here. And those papers didn't draw much attention. Yes?
As far as radioactive dating goes, that's, I believe, the vast majority of what public schools kind of rely on for their dating metrics. And so I was just wondering, I've seen other things like by Ken Hogan and whatnot about basically how there's been live snail shells dated at 13,000 years old and whatnot. So I don't know if maybe you could speak to that a little bit. Maybe if you've experienced maybe some of that in your own kind of testing.
There are anomalies that are even recognized by the evolutionist community as far as certain samples that you can't really test accurately because they give what would be clearly bizarre results. And that's why they come up with things like parentless argon. You know, when you date lava just extracted from a recent volcano and you get a 300,000 year age to it, you know, well, there's a problem here. Well, that's parentless argon. Carbon-14 on the shell of certain mollusks, you know, it dates at 1,000 or more years, but it's still alive. See, well, that's an anomaly, that's a problem. Typically what happens that I've observed anyway is that tends to just get dismissed as just kind of an anomaly. We don't really, it doesn't make that much difference. Now, I'm reluctant to be very happy with that answer because clearly that's pointing out that you got a problem, because how do you know that this problem doesn't extend into other samples where they're not alive? How do you know that it's, you know, if I'm dating a lava flow that I know is 2 years old and I get a 300,000 year date to it, if I'm dating a lava sample that I don't know how old it is and get a 300,000 year date, how do I know that 300,000 year is right or not? See, and that becomes, to me, that becomes one of the major problems in radiometric dating is when you date known samples and you get anomalous dates, then how do you know that it's going to work for unknown samples? That's the whole point of the dating process. So in many ways, one of the jokes is you have to date the sample in order to date the sample. And I'm not talking about taking it out for dinner either. I'm talking about, you know, you have to have an estimation of how old you expect it to be to know if your radiometric measurement is giving you an accurate measurement. And that's been a criticism of creationists leveled for years that again is often just ignored. Well, you dumb creationists, you don't understand anything. And that's where I'm saying that the dinosaur tissue is now providing an external challenge that has nothing to do with the assumptions that radiometric dating is based on or the assumption of sedimentation or anything like that. We also have C14 coming into the picture, and I didn't talk anything about C14. But if you know about C14, you recognize that with a 5,700 year half-life, you can't use C14 to date a fossil that you're claiming is 70 million years old because it would be all totally decayed out. And yet we find C14 in these fossils. C14 is consistently found in all fossils all across the geologic spectrum. Now, how in the world does that happen? Well, it's contamination is what the argument is. Two problems. First off, contamination never would behave in such a manner because contamination reveals itself by being very inconsistent. It'll be high in this sample, low in this sample, zero in this sample. But when you find that it's very consistent from location to location, fossil to fossil, geologic location, geologic position, that's not contamination. And also, theoretically, the C12-C13 ratio should give you an indication if you have contamination or not, and that we're not seeing the appropriate change in the C12-C13 ratio that you would expect if these fossils are contaminated with C14 should indicate also this is original indigenous C14, not contamination. And I go into more detail of that in my book, Echoes of the Jurassic.
I was actually going to ask about carbon dating, but I mean, I can still take a question.
Okay.
I was wondering if the variations in environmental factors causes carbon dating to be inaccurate in a second because of how volatile is the degradation of it in different temperatures? Is it like one of those more stable matters that there's not much in 70, 90, or is it pretty volatile?
I'm, again, probably not the best one to give you the complete answer on that because I'm not a physicist. And so if there are some factors that can influence carbon-14 degradation, I'm not one that can go into great expanse on those. But one thing I can tell you is with carbon-14, because it's such a short half-life, it's going to be much less affected than those that have a longer half-life. Because see, it would only take, all you need to, if you have a radiometric system that has a half-life of, let's say, 100 million years, 1%, how much does that throw it off? 10%, how much does that throw it off? You see, so a small percentage error throws it off a lot more than with carbon-14, which is much shorter. Also, one of the arguments when we're talking about carbon-14, because fossils tend to have a carbon-14 date of 35 to 45,000 years, then the evolutionist community say, well, see, that still doesn't fit your time frame, so that proves it's contamination. But see, that's imposing their model onto our model. Because our model says that at the time of creation, there would have been 0 carbon-14, because carbon-14 is a product of reactions in the atmosphere. So there's no reason to assume that the creator would have made carbon-14 to begin with. So if you start out with 0 carbon-14, at the time the dinosaurs would have been buried, the time other fossils would have been buried, the level of C14 would have been much less than it is today. Therefore, starting out with less, they would give an older carbon-14 age, but it actually is totally appropriate based on our model. Inappropriate to their model, but see, they can't apply their model to our model. Like, let us have our model, you guys have your model. And that's why you would get an age of 35, 40, 45,000 years, because they didn't have as much carbon-14 in them to begin with. And that fits, that's totally appropriate. Yeah.
From somebody who's in the battle, what could we do to help in the process?
Well, my first answer is going to sound kind of self-serving. Because research is expensive. You know, when I used to work in universities, it was well recognized. You put science on the front of any product and it goes up 10 times fold in price. I mean, research is expensive to do. And so just because we're working for a nonprofit, and sometimes we put the non in nonprofit. But anyway, it doesn't make it that suddenly research isn't less expensive. It's just as expensive for us as it is for the evolutionists. You know, we all are looking for money because it's expensive to do. And so, if nothing else, certainly financial contributions go a long way. But other things you can do, obviously, is you can, you know, buy books. You can educate yourself. You can talk to other people. You can buy videos. You can pass. We have people who that's their ministry. They buy videos and pass them out. They buy books and pass them out. See, so if nothing else, that can become a ministry. If you want, clearly. There's a college right down the road. I hear it's not too bad. It's one of those Big Ten schools, so, you know, anyway. All right, you can go to college and start getting degrees and join the fight from my end. And there's a lot of other things you can do clearly as well. Just spread the word, talk to people, challenge people. Write your congressman. Write letters to the editor. You could, depends on how much time you want to waste on Facebook, obviously, because some of it can be a big waste. But a lot of times on the internet, people will make a lot of comments that other people read, and if there's no response to it, they think, well, that must be true, and it's not. It's nonsense. And so sometimes by responding, at least that gives people an ability of seeing that there is a response. There is a counter-argument.
That might be a good place to quit. Thank you very much. And I think the